I'd like to begin by saying just how fortunate I know I was to begin my career by training at the LMB. It's literally no exaggeration to say that my time there totally shaped me, both in science and in many other aspects of my life. So how did I get to the LMB from the other side of the world? Well, during my undergraduate degree at the University of Melbourne, I'd fallen in love with DNA and molecular biology. So one day I just sat down and I wrote a letter to Francis Crick, asking him whether I could do my PhD with him at the LMB. And quite astonishingly, he said yes. Looking back, I can only marvel at both my naivety and audacity. So I arrived in Cambridge in September 1966, funded by a British scholarship. Thank you very much, because at the time, the only Australian awards, and there were precious few, were designated men only. I still vividly remember that first day, walking up the stairs in the old building, of course, past the giant model of TMV to be interviewed by Francis Crick and Sydney Brenner. My heart was literally in my mouth. In short shrift, they assigned me a project, sequencing mTRNA, and a supervisor, Brian Clark. I was very lonely at first, but gradually came to enjoy my life in Cambridge, singing in comes, training with the first mixed bumps crew, and taking exhilarating train rides to London to go to the opera and the art museums. And of course, next slide, please, getting to know US postdoc Jerry Adams on the left there, who stole my heart and became my scientific and life partner. Many of the friendships that Jerry and I made at the LMB have endured for decades, particularly those with Joan and Tom Stites, but also with Mark Bretcher and George and Susan Brownlee. And of course, the network of contacts that we all made there opened many doors through our subsequent careers. I found that life at the LMB was both daunting and exhilarating. Literally, for the first time, I realized that science was a vocation, not just an occupation. Every day I was exposed to penetrating minds, passionate about unlocking the molecular secrets of life. And of course, being the first in the world to do so. I learned that success comes from asking big questions, and that answering these big questions often depends on new technologies, which were in plentiful supply, of course, at the LMB. Next slide, please. I was fortunate to learn Fred Sanger's then new RNA sequencing methods, and this gave me not only my PhD with the sequence of mTRNA, but also my postdoc in Geneva, where Jerry and I sequenced regions of R17 phage RNA as a model messenger RNA and discovered a high degree of unanticipated secondary structure. Next slide, please. After Geneva, Jerry and I took a big gamble and we decided to set up our own lab in Australia. Of course, that was my fault, but I'm forever grateful for Jerry to agree to trial this. We looked for a place that had a similar scientific atmosphere to the LMB, and we chose WEHI, the Walter and Eliza Hall Institute of Medical Research, which had become an internationally famous immunology center under Nobel laureate Macfarlane Burnet and his successor, Gus Nossel. Next slide, please. Our first success in Melbourne, just before our first 3-year NIH grant ran out, was the discovery of 5' caps and other modified nucleosides in immunoglobulin messenger RNAs and other eukaryotic messenger RNAs. And it just happened that Fred Sanger visited our lab in Melbourne during this period, and we were really thrilled when we showed him our data anxiously, and he offered his usual very understated approval. I think that looks about right. Little did we know that nucleoside-modified RNA would eventually drive a revolution in vaccine development. With the advent of recombinant DNA technology in the mid-70s, it became possible to pursue our original goal in Melbourne, the genetic basis for antibody diversity, a topic for which we'd been well prepared by rubbing shoulders with Cesar Milstein at LMB. Next slide, please. We cloned immunoglobulin genes and made significant contributions to understanding their organization in the germline and then their reorganization in antibody-producing B cells. Our most significant discovery was that Ig gene rearrangement involves DNA deletions, a process still unique to lymphocytes. In the early 80s, we switched to cancer genetics, inspired by Bishop and Barmas's discovery of cellular oncogenes. And this led us, next slide please, and others independently to discover that chromosome translocations in human Burkitt's lymphomas are caused by the juxtaposition of the oncogene MYC near Ig gene enhancers, thereby promoting ceaseless division, as you can see on the diagram on the left of that slide. Enabled by a collaboration with Ralph Brinster and Richard Parmiter, who pioneered transgenic mouse technology, we engineered mouse to model the MYC translocation and were totally blown away by the fact, but when we got the first phone call from Philadelphia, every one of these mice developed lymphoma, a stunning proof that the MYC translocation was the fundamental cause of Burkitt's lymphoma. And of course, this acted as a precedent to hunt for other oncogenes activated by recurring chromosome translocations in leukemias and lymphomas. Next slide, please. The next translocation that we studied was the 14-18 translocation in follicular lymphoma, another type of disease, and the gene that was linked to the Ig gene locus in this case was given the name BCL2 for B-cell lymphoma gene number 2, but its sequence didn't reveal anything about its function, which remained a mystery for some time until David Bowe, a PhD student in our lab at the time, found that, in fact, when he overexpressed BCL2 in cytokine-dependent cells, shown on the right, instead of the cells dying in the absence of that cytokine, they remained totally viable. They didn't proliferate anymore, but they stayed viable. And we were totally blown away by this finding. At first, we thought it was a bit boring, and then we realized as we thought more about it, that this was a really fundamental discovery. First of all, it told us that cell survival is controlled separately from cell proliferation, which was not at all clear at that time. We generated more transgenic mice. Next slide, please. And we crossed them to our MYC transgenic mice. And you can see with the yellow curve there that the mice that carried both the BCL2 and the MYC oncogene developed tumors far faster than those carrying just the MYC oncogene. So we had proven by this experiment that BCL2 promotes oncogenesis, and that from other work that we did at the time, and others did as well, particularly Gerard Evan, we found out that the reason that BCL2 collaborates with MYC in oncogenesis is because it inhibits the ability of MYC to induce apoptosis when cells are stressed. But the really important fundamental finding from this was that this was the first demonstration that inhibiting cell death is a major step towards tumor development. Now a totally accepted concept, but totally new at the time. Okay, well, it turned out, next slide, please, that BCL2 has many homologs. Some of them, like BCL2, also promote cell survival, the blue ones at the top there, but others, although they're very similar in sequence and in structure, actually, instead are absolutely essential for cells to die. They're called BACs and BAC. And then there's a more distantly related faction called the BH3 only proteins, because they share just this particular motif, the BCL2 homology region number 3, with all of the other family members. And it's the BH3 domain of both the BH3 only death proteins and the BACs and BAC death proteins that's essential for their pro-death activity. So next slide, please. Gradually, the field worked out over time that the BCL2 family interact with each other at the mitochondrial outer membrane, and that together they act as a life-death switch in cells. In healthy cells, the guardians, BCL2 and its closest homologs, prevent activation of the pro-death proteins, BACs and BAC. But when cells undergo stresses, the cell synthesizes or releases the activity of a number of BH3 only proteins, which then bind tightly to BCL2, the pro-survival proteins, preventing them from inhibiting the death duties of BACs and BAC. And the result is that BACs and BAC form pores on the outer mitochondrial membrane, and that allows the leakage out of the mitochondria of cytochrome C, which leads to the activation of caspases, which demolish the cell by apoptosis. Okay, so this is in a nutshell how the BCL2 family works. And it was this increased knowledge about the molecular details of the life-death process that has led to a major new class of cancer drugs, which we call BH3 mimetics. And as you can tell from the name, next slide, that's because these small molecules mimic the binding of the BH3 only proteins to a groove on the pro-survival proteins. So here in this picture, the pink is what where the BH3 domain binds from a BH3 only protein. And in the yellow and aqua, you can see this small molecule, a BH3 mimetic, which has been developed. The first bona fide BH3 mimetic by Abbott Laboratories. And I have to say this was a Herculean effort of chemistry informed by structural biology. This particular BH3 mimetic, the first one, binds very strongly to 3 of the pro-survival proteins, BCL2, BCLXL, and BCLW, but not to MCL1 or A1. But it had an unfortunate side effect when it was tested in the clinic. It provoked neutropenia, and this was due to an on-target inhibition of BCLXL in platelets, because it turns out that BCLXL is absolutely critical for the survival of platelets. Next slide, please. Well, we were in Melbourne very engaged with our collaborators in trying to develop our own BH3 mimetics, and we entered into to cut a long story short, we entered into a research collaboration with Genentech and AbbVie, or Abbott Laboratories, and together we developed a BCL2 specific BH3 mimetic, which came to be called venetoclax, and that is now FDA approved for treatment of chronic lymphocytic leukemia and acute myeloid leukemia, and it's in many trials for many other kinds of cancers. It's been a very, very exciting journey. So I just wanted to finish off, well, for a minute, turn to another facet of my career. Next slide, please. And that is in 1996, I was asked to become director of WEHI. And first, I was very hesitant to take on this role because my own science was going very well, and I knew that this would present a major challenge trying to be a director as well as a research scientist. But I thought, on thinking about it, I felt very grateful to WEHI for enabling me to pursue a successful scientific career in Australia, which was not at all obvious at the time. So I did accept the challenge. and built WeHire bigger and stronger. But I wanted to emphasize here that many of the decisions that I took during that period continued to be influenced by my experience at the LMB. For example, the introduction of structural biology, which is now one of WeHire's major assets, was absolutely key to the development of venetoclax and the work that we did on that at WeHire. So in closing, like most of us here today, I'm often asked for career advice by young women. And I always go back to my LMB experience for answers. I tell them, follow your scientific dreams, train with the best, only tackle important questions because life's too short to be collecting scientific postage stamps. Acquire a new advanced technology. This will open up your career opportunities. Work very hard, but don't neglect other life experiences. Very importantly, choose a supportive partner and take up opportunities courageously. But above all, remain passionate about science and discovery. This is the energy that will sustain you through the highs and lows, not only of science, but also of life. And with that, I'd like to finish and thank everyone very much for their patience during this rather unusual presentation with all the interruptions. Thank everyone who helped make it come to pass. And especially I'd like to thank Marianne and Kathy for inviting me to be part of this historic meeting. And thank you, LMB.